Hello students, once again welcome back to biology class. Myself Dr. Raghavendra here. Well, in the earlier class we discussed about the certain introductory points related with the biomolecules. So basically the definition of biomolecules, it says that the chemical molecules produced as a result of metabolism in the living cell. We call them as a biomolecules. Fine. Biomolecules include the proteins, carbohydrates, lipids, nucleic acids. Variety of certain chemicals, they may be acting as a cofactors. Biomolecules are not only always made up of the organic compounds, they may be having the inorganic component also in them. Fine. So we discussed about basically the biomolecules which can be there are lots of similarity between the living as well as non-living things by chemical nature. If we consider the human body and earth crust, yes, we find the similar kind of chemicals. What is that? Nitrogen, okay, hydrogen, oxygen, magnesium, calcium, carbon, sulfur, sodium, silicon. So variety of chemicals which are there in the earth crust, yes, that can be seen in our body also. We have discussed about that. And there are certain chemicals, yes, we call them as exclusively inorganic chemicals. They can be such as it may be the sodium, it may be the potassium, it may be the calcium, magnesium, okay. So thus there is again water and certain compounds they are going to form like NaCl, CaCO3, MgCl2, SO4, PO4, yes, they are going to form, and all those are required for to run number of variety of metabolic activities inside our body. So we understand there are lots of similarities between the living and non-living by the nature of chemical or component of the chemicals. How we are able to analyze these chemical component of the living cell or tissue? So take the tissue, yes, you are adding a trichloroacetic acid, grind the tissue, addition of TCA makes the cells to disintegrate. It mainly digests the collagen tissue also, and then cells get separated. It has become very easy to break the cell now. Cells are broken, all cellular content, cytoplasmic content comes out, and then you apply either centrifugation or you apply the filtration mechanism. The content is called as a slurry. When you filter it, you will get the acid soluble pool that has a protein, carbon and certain amount of lipids also. Acid insoluble pool, yes, you may find the inorganic substances also in them, as well as you will find a good amount of lipids also in them. That is called as acid insoluble pool. Further, in the test tube, what you have collected all the acid soluble pool in that proteins are there, carbohydrates are there, nucleic acids are there. You conduct variety of chemical assay or test, you will analyze, okay, the concentration of so and so particular chemical per gram weight of the particular tissue, you will analyze. This is done in the biochemical laboratories. Now when we talk about basically what we can say, the mass spectroscopy, yes, let me tell you how actually it is done. Mass spectroscopy is a technique of analyzing what we can say that the chemical nature of a compound. Yes, you have collected some compound but you don't know what are the different components in that. So that is mainly done by what we can say that the mass spectroscopy. So let me tell you how actually they do. So it is also called as atomic absorption spectroscopy. Yes. What they will do in this method, the tissue or the chemical compound, yes, it is now burnt by exposing to the flame or fire. It is burnt. After burning, whatever the organic tissue or the chemical compound, what you will get? You will get the ash. So this ash is compressed. Made into a pellet. Yes. So what they do? This pellet is now what we can say, it is the chemical pellet. Yes. What we can say that chemical pellet. Then there is what we can say light rays. So UV light rays will be passed through this. Okay. So mainly the ash pellet. Then the rays are passed through that. So what will happen further? It is connected actually with the system, the computer, and then it gives the peak values. So what the peak value says, let me tell you here. So it is a compound, what we can say that concentration. Okay, so the y-axis is a concentration, it is mainly OD. Okay, optical density we call it as. Every chemical has its own, what we can say, absorption maxima. Every chemical absorbs the particular range of wavelength of light to the maximum level, absorption maxima. So what we can say that it will show certain peaks like this, it may show certain peaks like this. Okay, yes. So what we say mainly, these are, okay, the C1, C2, C3, C4. So it is mainly OD means optical density. Optical density. Okay. So C1 to C4 are the components. Okay. Yes. Components mainly having different absorption maxima. Based on this, what they do, this whatever the ash pellet or the compound has so many components, they will analyze. Further they go for the molecular structure analysis also. This result helps in the further molecular structure analysis also. What we call it as mainly the atomic absorption spectroscopy. Okay, I hope the results, yes, the results used to construct, okay, yes, molecular structure and formula of the compound. Okay, this is what we talk about mainly, how actually why the chemical or the tissue or the material is burnt and converted into ash, because that is the ash compressed and made into pellet, right? So this hard substance or pellet fixed in the device, and then the rays or light rays are passed through that, and it is connected to the computer, and the computer will show or exhibit different peak levels of the different components of that compound. So here the y-axis represents concentration. Let me mention clearly, concentration. Fine. So thus these are the different peak values at different optical densities, indicating the details of the particular, yes, components in the particular compound. This is what we talk about mainly, analysis of biomolecules. Well, so let us go ahead with the primary and secondary metabolites. We need to understand what is primary metabolites and what is secondary metabolites, and there are number of secondary metabolites also, and their use we are going to discuss. Fine. So that also we are going to study now, primary and secondary metabolites. After that, yes, we are going to discuss about the, yes, structural units of mainly biomolecules. So let us understand now primary and secondary metabolites. So the biomolecules is a quite interesting chapter, and its study is mainly gives us a different dimension about the biology. Okay, so we need to understand each and every point very slowly and thoroughly. Okay, fine. Let me go ahead with now the next. Yes, we are going to discuss the primary and secondary metabolites. What are these metabolites? Yes, anything that is resulted from the cell metabolism, we call the metabolites. So we eat food. Food is a complexion of protein, carbohydrate, lipid, and food is broken into, yes, amino acids, carbohydrates like sugars, and then the fatty acids. Yes, this is what we say the metabolites of those biomolecules. So let us understand what is mean by primary metabolites. Primary metabolites, metabolites, and secondary metabolites. Fine. So when we say primary metabolite, they are produced inside the cell abundantly. Produced inside the cell. Yes. Secondary, produced inside the cell and secreted out. Okay, the first difference. Secondly, we can say that so play important role in metabolism. Okay, yes, function not clear. These required for the cell compulsorily. Yes, requirement may be or may not be. Required may or may not. Means the cell can survive without secondary metabolites. And then we can say that the primary metabolites basically, so they are examples includes proteins, carbohydrates, okay, lipids, nucleic acids. Okay. So mainly secondary metabolites includes mainly it can be pigments, okay, it can be alkaloids, okay, then it can be the drugs, it can be the toxins, etc. Many are there. So the secondary metabolites are produced and they are secreted out. The functions are not much clear. Cell may not be in need of those secondary metabolites, but primary metabolites are produced abundantly inside the cell. Cell only will utilize them and they play key role in the overall maintenance of the cell homeostasis or cell metabolism. So looking into yes, number of different kind of yes examples are there about secondary metabolites and their even the use applications we can say or functions we can say. Let us understand those types of secondary metabolites. So and their importance or examples. Types of secondary metabolites. Okay, and then examples. There are number of examples under the secondary metabolites. So as the first one may be the pigments. Pigments that includes carotenoids as well as anthocyanins. Yes. Then alkaloids are there. Alkaloids, mainly morphine and codeine, etc. Okay. Then terpenoids. Terpenoids are there. That includes monoterpenes and diterpenes. Okay. Then essential oils are there. Best example, lemon grass oil is one of the important example. Then lectins are there. Yes, concavalin A we call it as Okay, yes. Then there are number of drugs or the medicines that can be, yes, vinblastine, vinblastine and curcumin. Other than this, we can say that mainly what we can say polymers, polymer compounds such as it may be rubber, okay, it may be cellulose, it may be the gums. These are all the polymers, okay. Then there is what you can say toxins. So toxins are the poisons produced by number of plants. What are those toxins? They are called as abrin, okay, abrin as well as resin. So these are the different types of mainly secondary metabolites produced by the different kind of plants. We can say that yes, in the plants secondary metabolites are abundant. So when we say pigments, it may be the chlorophyll, it may be the carotenoids, the carrot, anthocyanins are there. These are all the pigments, yes, may be essential for number of other metabolic activities, but produced by certain cells, and then the color is exhibited. Then alkaloids, mainly the drugs like morphine, okay, it is also used as a painkiller, morphine, but too much of intake causes addiction for the morphine, even the codeine also. So these morphine and codeine are the drugs which are acting as a sedatives or the painkiller, but when they are utilized in excess, that causes the addiction. And then terpenoids, monoterpenes and diterpenes are the kind of chemicals can be used in the wood paints, okay, monoterpenes and diterpenes. Then essential oil, lemongrass oil is there, even castor oil, even groundnut oil, variety of yes, plants, seeds, they do have the different kind of oil can be extracted. Even from the eucalyptus we can extract the oil. So these are all called as essential oil, which are not required for the plants, but they are aromatic compounds that can be utilized for some other purpose like a medicine. So lemongrass oil that can be used mainly in the what can say the relieving the pain, or mainly to reduce the joint problems also. Lectins, concavalin A, it's a compound that can be acting as a immunoregulator. It is a compound that can be acting as a immunoregulator. Yes, it is known to initiate or enhance the activities of the T lymphocytes, and it will boost up mainly the immunity. So concavalin A. And then drugs like vinblastine. Yes, vinblastine is actually isolated from a plant called as Vinca rosea. Vinca rosea, batalahu, what we say in Kannada. So Vinca rosea or vinblastine used to treat the cancers of the testicles, cancers of the variety of organs. Yes, vinblastine mainly. Vincristine is a active ingredient, and vinblastine is isolated from the Vinca rosea, the plant Vinca rosea. And then curcumin, yes, curcumin or turmeric we say, turmeric. So curcuminous longa we call it as the turmeric, scientifically called as curcuminous longa. And then from this curcumin and turmeric powder can be utilized as an antibacterial agent or antibiotic. It can be used, yes, it can be used in the food also, and that is very important consuming the mainly turmeric powder, because yes, in a limited range that increases our immunity and reduces the internal bacterial infections. Well, rubber, cellulose, gums, these are not the plants producing for their own sake. They are producing, releasing it out. Human can utilize them. Okay, and then so rubber and cellulose. Yes, in the cotton, what we find the fibers are all made up of cellulose only, gums. And then mainly there are number of plants, their gums are taken and then used to make up the sweet or laddus. Okay, well, toxins such as abrin from a wild, what you can say, bean, resin, Ricinus cuminis is a plant called as castor. So the seeds of the castor and you are taking in the mouth, crushing, suddenly it can cause the death also, and because they are so much toxic. So castor seeds, so ricin and abrine are the toxins. These are all what you can say secondary metabolites produced by the plants, mainly for their own defense, and but there may be what you can say for the human are capable of utilizing them in an effective way and making in a different forms. Well, this is all about what we have discussed, the secondary metabolites. Now let us go ahead with the biomolecules in detail. Yes, what we will be discussing about the mainly the biomolecules in detail as such. In the biomolecules, yes, we need to understand basically the biomolecules based on their molecular weight, how they are classified. Okay, we call them as bio micromolecules, bio macromolecules also. And then biomolecules, yes, they are not all of a sudden they are made just like that. They are made up of different kind of structural units. So we will be discussing about the most important structural units of the proteins, carbohydrates, as well as the lipids and nucleic acids. Okay, and their importance also we will discuss. Now let us understand. So basically biomolecules. So biomolecules we can say based on molecular weight, the biomolecules are of two types. We call it as a bio micromolecules and bio macromolecules. So how biomicromolecules? Examples includes mainly, yes, biomicromolecules molecular weight between 18 Dalton. So generally the proteins are measured in terms of the, yes, unit called as Dalton. 18 Daltons to 800 Daltons. Okay, this is biomicromolecules. So biomacromolecules, you can say that their molecular weight between, we can say that 800 Dalton, okay, 800 Dalton above, so 800 Dalton or 1000 Dalton, yes, above, and maybe beyond what they can be, beyond 10,000 Daltons also. So such compounds we call them as the biomacromolecules. Biomicromolecules, they are from acid soluble pool. Okay, acid soluble pool, or we can say that cytoplasmic content, we can say. Example includes proteins, carbohydrates, and even what we can say that nucleic acids. Yes. So here these biomacromolecules from acid insoluble pool. Okay, so what we can find in the acid insoluble pool? They are the lipids. Okay, but molecular weight not more than 800 Dalton. Thus, so lipids are not compulsorily macromolecules. Okay. So acid insoluble pool, that means mainly cell membrane and organelle membranes. Okay, from there it has been taken. What is this biomolecules acid soluble and acid insoluble means? Yes, we have discussed in the earlier class that so when we crush the tissue, okay, plus PCA, and then we get the filtrate. So here we find acid insoluble and here we find acid soluble pool. Okay, so this is what we say mainly the types of biomolecules based on their molecular weight. So this is what we talk about. Yes, bio micromolecules as well as bio macromolecules. And basically bio micromolecules are proteins, carbohydrates, lipids. Macromolecules mainly the lipids, but not a compulsorily the macromolecules, because they may be acid insoluble but their molecular weight never go beyond the 800 daltons. So based on molecular weight, lipids also micromolecules only. Fine. So when we say that mainly the biomolecules in a cell, they can be of a different percentage value. So different percentage value they exhibit. So such as, let me tell you percentage value of biomolecules. So biomolecules, okay, percentage in cell. Biomolecules as a percentage wise we want to measure means it may be definitely the water as such, may not be the biomolecule. Yes, definitely biomolecule. It will be 70 to 90% in a cell. Proteins as such 10 to 15% in the cell. Then carbohydrates that form mainly 3% in the cell. Lipids 2%. Ions forms 1%. And then nucleic acids, sorry, they are again mainly nucleic acids. Nucleic acids that includes RNA and DNA, that forms 5 to 7%. So these are the percentage value of biomolecules within the cell. This you need to remember. So this is what we talk about mainly the biomolecules based on what we can say the molecular weight. They are of different type, and then they can be like acid insoluble, mainly the lipids, acid soluble, protein, carbohydrate and lipids. Yes, the molecular weight of acid soluble pool or the what you can say bio micromolecules, their molecular weight in between 18 dalton to 800 dalton, whereas macromolecules, yes, their molecular weight will be in between 800 to 1000, sometime beyond 10,000 dalton also. Such a large bio macromolecules we can find. So a cell, when we talk about, yes, a cell has lots of most important biomolecules, thousands of metabolic reactions will be happening, and if we keep on analyzing and isolating, yes, we may find thousands of biomolecules, but all of them in one or other way, the combination of carbon, hydrogen, oxygen and nitrogen only, they are all made up of. So all biomolecules made up of, we can say that all biomolecules have mainly what we can say carbon, hydrogen, oxygen, nitrogen and phosphate groups. Okay, we will be discussing in detail about mainly the structural units of these biomolecules in the next class. Till then, take care, study well. Thank you very much.